Hi, my name is Hannah Petronic. I am a senior biochemistry major at West Virginia University from Wheeling, West Virginia. I do research in the Casson Lab within the Division of Plant and Soil Sciences, and the title of my research presentation is Exploring Species Boundaries for Neonectria magnoliae. We are all familiar with getting horrible canker sores in our mouth, and while trees happen to get something called cankers too. Cankers on trees are localized necrosis of a trunk or branch, which in some cases can girdle and eventually kill the tree. Cankers like these are often caused by fungi or bacteria, taking advantage of already weakened spots on a tree. We can see some examples of different types of cankers to the right of my slide here. Fungal pathogens play a huge role in forest health and tree mortality. In my research, I study fungi within the family Nectriaceae, in which there are many species that cause cankers on numerous hosts. You may have heard of beech bark disease, which has been in the United States since the 1800s and was first found in West Virginia in 1981. Beech bark disease is a canker disease that affects American beech and is caused by both Neonectria faginata and Neonectria datisma. We can see a photo of beech bark disease down here in the bottom left. From 2016 to 2019, Cameron Stodder, a previous grad student in our lab, conducted a survey looking for Nectriaceous fungi throughout the Appalachian Mountains. While beech bark disease is extremely prevalent, he also came across many other similar pathogenic fungi on a wide variety of hosts. That leads us to ask several questions. If the forests in West Virginia are so well studied, how did we not encounter these pathogenic fungi before? What sort of hidden diseases have we uncovered? And what other canker-causing fungi are out there? In Stodder's survey, he came across Neonectria magnoliae in Furneaux Experimental Forest and Snake Hill. Neonectria magnoliae is a weak plant pathogen found on both Fraser magnolia and tulip-poplar. Like the fungal pathogen and beech bark disease, along with other nectriaceous fungi, Neonectria magnoliae produces bright red fruiting bodies, known as parathecia, which can be seen in the two rightmost images. Neonectria magnoliae causes perennial target cankers, which can eventually wrap themselves around the main trunk or branch of a tree and kill it. Milder winters due to climate change may be the reason that weaker pathogens like this one are becoming more prevalent in our forests. As Fraser magnolia and tulip poplar are at two very different elevations, we have been led to believe that Neonectria magnoliae might actually be two different species. Even though it is a weak pathogen, investigating the species boundaries can help us prepare for future epidemics of this fungal pathogen and help us to better control it. In fungi, we classify a lot of things based off of their morphology. One morphological characteristic we look at is conidia, which are the asexual spores of fungi belonging to the phylum Ascomycota. As mentioned in the previous slide, we have some evidence that suggests that Neonectria magnoliae might be two different species. In addition to having two different hosts at different elevations, Neonectria magnoliae from each host is slightly different in their morphology. For Neonectria magnoliae isolated from tulip poplar, we only see microconidia, which can be seen in the bottom right image here. Whereas Neonectria magnoliae isolated from Fraser magnolia exhibit both macro and microconidia. This evidence supports our hypothesis that Neonectria magnoliae is actually two cryptic species. Stodder's comprehensive survey also unveiled the evolutionary relationships among nectriaceous fungi. In this phylogenetic tree on the right of my slide, we can see the clade where Neonectria magnoliae falls. Although both strains from Fraser magnolia and tulip poplar are closely related, it appears that isolates from Fraser magnolia are slightly different genetically from tulip poplar isolates. This may indicate a recent species divergence. Our ultimate goal with this research is to delimit the species boundary. My own samples were collected throughout the state of West Virginia in places like the West Virginia Botanical Gardens, Audra State Park, and Godnier Knob. After sequencing my samples from tulip poplar and Fraser magnolia, we can clearly see that they are different genetically. Tulip poplar strains branch off into clade 1, while Fraser magnolia strains branch off into clade 2. An intriguing result is that Neonectria magnoliae samples isolated from tulip poplar from the West Virginia Botanical Gardens right here in Mon County seem to be slightly different genetically from other tulip poplar strains. In addition to this, it was found that samples isolated from alder came back as Neonectria magnoliae. This is a fascinating discovery as alder is not of the family Magnolia, whereas Fraser magnolia and tulip-poplar are. This just goes to show the wide range of biodiversity we have here in West Virginia. In order to further investigate species boundaries, we have set up cross-pathogencity assays in WVU's research forest. Due to the abundance of tulip poplar in this area, we decided to start with only a cross inoculation of tulip poplar instead of including Fraser magnolia as well. To perform this cross pathogenicity assay, we took Neonectria magnoliae strains from tulip poplar and inoculated 10 healthy tulip poplar trees in the university forest. Likewise, we took Neonectria magnoliae strains from Fraser magnolia and inoculated an additional 10 healthy tulip poplar trees. We are currently at 3 months post-inoculation and have yet to see any evidence of canker formation, but we hope that this assay can help us to better understand both the pathogenicity of Neonectria magnoliae and if the Fraser magnolia strain is able to colonize tulip poplar. As we move forward with this research, we will continue to keep an eye on our cross-pathogenicity assays and eventually include Fraser magnolia. In addition, we will be performing these inoculations with alder as well. Because alder was a non-magnolia host, we will be looking for Neonectria magnoliae elsewhere. Our search will continue for Nectriaceous fungi both within West Virginia and outside of West Virginia. Thank you for listening. Please let me know if you have any questions or comments.